Okay, so this is where we are just now. Questions. So viruses can be cultured using the same method as bacteria and fungi. Why is this wrong? The answer is false. Why? Okay. So some of you may be scoring 100%. The answer is false. What type of cell can infect virus? What type of cell can viruses infect? It's actually all of them. How do viruses reproduce? They take over the living cell's machinery and not the rest. Protein shells are also known as capsid. This is a shell shape. If you have a closer look, they are rods. Okay, long shaped rods. And the cell arrangement for this one, this is Sakina, and this is a group of 8. So not tetrad, not strepto. Strepto is in chain form. Staphylococcus is grape-like. Which of these would you not expect to see in bacterial cell? So bacterial cell, they are prokaryotic cells, so there's no nucleus. They have nucleus, but not nucleus. Bacterial cells are prokaryotes. Letter A. Letter A, this is the flagellum. So this tiny little spike here, they are the pili or pillars, singular pillars. And if I look at this one, what is correct for this cell here? So they appear as red color, so they are gram-negative cell. So gram-negative cell, and in terms of the arrangement, they are streptobacilli. So they are short, they are rods in chain. Which part of the bacterial cell wall holds the same, which is a peptidoglycan, if you recall, which is applied last in gram staining, safranin. So if you remember the order of gram staining, it will be crystal violet, iodine, decolorization using alcohol, and then safranin, which is red. So viruses are cell, which is not correct. They are acellular. They are not really cell. They are only living when they are attached to whole cell. Otherwise, outside cell, they are non-cell, acellular. A bacteriophage is a virus that enter a bacterial cell. So bacteriophage is also one that infect viruses. Bacteria. Antibiotics kills viruses, which is not correct. Antibiotics only kill bacteria. So if you have viral infections, you use antiviral. Vegetative cells are much more sensitive to heat than spores, which is true. Spores are the formation of, which is a specific structures that's present in fungi. Vegetative cells simply means that they are actively growing, but spores, endospores, and bacteria are not actively growing. Chemical agent that kill the microorganism is commonly applied to substances used on inanimate objects are known as disinfectant. Antiseptics are those that are used on living tissue. Sanitizers also used on living tissues. Antibiotics is not really used on, it is for us to consume. So all other answers, antiseptic, sanitizer, antibiotics are not correct. So which of the following is a characteristic of sanitizer? Reduces the microbial population to safe level. Low temperature reduces the activity of microbes by reducing the chemical reactions and action of enzyme. Aerobic bacteria require oxygen to survive, in opposed to our anaerobes. Anaerobes, for example, is a bacteria that do not require and do not like oxygen. So when streaking a plate, the loop should be flame at the beginning and in between each zone. So if you do a streak plate, you will have 4 quadrants. At each quadrant, you will have to flame the loop to kill all the bacteria that's on the loop before you streak further. So that by doing that, you'll get small numbers of bacteria to form colonies on agar plate. So this is more technical. Question 21. Why is it important that petri dish are incubated upside down? So this will prevent moisture from condensing onto the agar surface. Okay, right. So I think most of you have done quite well. Let me see. So I will go on to the lectures. Introduction. Hopefully a quick one. hang on. Stop sharing. I will turn off my camera so later on, if you look at the lectures, you will not be seeing my face blocking the slide. Okay, all good, huh? So a very quick lectures on this. So the outline for the lecture today, we'll be looking at the definition of medical microbiology. Then we'll look at the past, what happened in the past in terms of medical microbiology, and we'll look at what is happening at the present, what are infectious disease as a leading cause of death in the present time. I'm sure you're already familiar with this. The COVID-19 pandemic has taught us big lessons in terms of how ready are we, how prepared are we against pandemic, and also a lot of the knowledge for science, life sciences are very important. And in terms of future, what is going to happen? What is the future directions in medical microbiology? So can one of you tell me what is this image in here? All these dots, what are these things? If you can turn on your mic, what do you think they are? Have a guess. What are these things? Anyone? Some cells, right? Oops, sorry. How did I accidentally turn this off? Some cells. What kind of cells are they? And what are these tiny little dense dots there? Anyone? Must be some bacteria or virus, right? No one? Okay, never mind. First lecture. Okay, all these tiny little dots here are actually the dengue virus that infecting and growing, replicating inside. So as you know, dengue virus and dengue infection is one of the most common infectious agent in Malaysia. Very common. The second type of common infection, what would that be? So there are many. If you're talking about bacterial infection, they can be contributed by bacteria, sore throat, for example. But having said that, viruses can also cause a throat infection, right? So dengue is one common one. Among the young children, hand, foot, and mouth disease is the other one. Okay, let's move on. So the objective for the lectures today is to be able to explain, for you to be able to explain the concept of medical microbiology in terms of the definition and what are the application of medical microbiology, and looking at the looking at the history, what is the historical impact of pandemics on human health and also society, and also to be able to explain the major infectious diseases that continue to cause significant mortality today, and also to identify future direction in medical microbiology. So again, this image here is taken from one of the websites. These round cocci, they are bacterial cells. They are attaching to a surface. So in terms of arrangement, they are like a little bit like grape-like structures. They form in clusters and sphere in the shape. In terms of the definition of microbiology, we are looking at the study of microbes. So microbes can include viruses, bacteria, fungi, and parasites in the order of the size of this microbe. So it is a study of microbes that cause illnesses. Illnesses referring specifically to infectious diseases and also known as communicable diseases. So non-communicable diseases are heart disease, diabetes. So these are not communicable. Communicable infectious diseases that can pass from one person to the other person, or we can get the infectious disease from the environment, from animals if it is zoonotic. And that is the definition of medical microbiology. So you will note that here I say microbes and not microorganism. So just now when we did the quizzes, there's one question whether viruses are cells or not. So because microbes encompasses viruses, viruses are not cell, they're not organism. So we say microbes to include viruses as a broader term. But if you refer to microorganism, that will not include viruses because they're not cell, they're not organism. Okay, the study of microbe, not just the study of microorganism. If it is a microorganism, we mainly refer that to bacteria, or we may be referring to archaea. Maybe from year one you remember archaea bacteria. They are also a unicellular organism, but they are not prokaryotes. Okay, so in medical microbiology, we study the biology of the microbe. We look at diagnostic method, we look at surveillance, we look at control and prevention and treatment of infectious diseases. So I'm sorry, why is it moving so fast? So you may also come across the term clinical microbiology. Clinical microbiology is a subset of medical microbiology. Clinical microbiology deals with the diagnosis of infections in patients. So for example, if you work in a clinical lab or path lab, Sunway Medical Lab, you only look at human samples that is who are suspected to have infectious disease, and your job is to diagnose whether the samples collected from the patients has got the microbes that potentially cause the signs and symptoms of that patient. So clinical microbe is a subset. Medical microbiology covers many more things, not just diagnosis, but also the study of understanding of the microbes, such as the life cycles, the surveillance of the infectious diseases, how do we prevent and control and treat the infectious diseases. Okay, so if you look at, so that is the definition and the scope of medical microbiology. If you look at the past, the history of pandemic, why is this important? So the understanding of past pandemic will help us to learn a few things. One of them is how diseases spread across population and how society respond in terms of medical response, social response, and politics response. And also, why is public health system and scientific research become necessary? So this is happening in the past. Learning from the past, we understand and prepare for future pandemics. So if you look at the history of pandemic, there are few examples. So we have Black Death that happens in the 14th century. We have the 1918 Spanish flu, and also the HIV and AIDS pandemics. Let me just read the message. I heard that there are a few. Oh, the screen is not changing. Can you let me reshare again? I'm already at slide number 4. There's some issue with this. Can you see now? Can you see me on slide number 6? Yes? Oh, the screen won't share when it's in full screen. Okay. In that case, I will just maximize this. Is that okay? That's not too small to see, right? Okay. So just now, when I was doing the sharing, you can't really see the previous slide. Okay. So why not I'll go this way so everybody will see, right? So this is what I have already talked about just now, the definition, and also why do we need to study what happened in the past? Okay, the history of a pandemic. So if you look at this image here, it is one of the images that records what happened in the 14th century when the Black Death happened. Those of you who are interested in history, you may have come across this. So Black Death is also one of the plague that happened that killed a huge numbers of European population years back. So it is actually caused by a bacteria that was spread by different animals. So this is what happens. A lot of people die, and it was predicted that it actually wiped out about 1/3 of the European population back then. So this is another image that shows what Black Death is. So Black Death, why did it get this name? Because when a person was infected by the bacteria, they will form all this bubonic plague, some forms of growth along the body trunk, especially, as you can see in the pictures here. And as time goes by, all these structures that grow will turn black color. So over here, you see 2 persons on the bed. We have that has a lot of bubonic that is growing on their body. So here you see one person sort of like praying to, in the image. So what happened is, because back then the cause of the infectious disease was not known, and it was believed that what happened then, a lot of people got infected and died. It was thought that it was a punishment of the god. So not only that it has medical impact like death toll, it also have some societal and some impact on the religion. So if you read further on Black Death, in different platforms, different websites, you will know that one of the things that happened in the 14th century following Black Death is also mass killing of Jewish people. It was thought that because, so what happened was, how did it spread? It started when a group of sailors or people who travel by sea, they came back, and a lot of them got infected. And these people who travel, a lot of them, not many of them were Jewish, and Jewish was thought that they were doing something behind that causes the pandemic. So a lot of people Jewish were killed back then. So there are a lot of history and a lot of stories behind the Black Death pandemic that you can have a read. I think it's quite interesting. And moving on, if we see the history of pandemic, you'll be able to see the original of this website and the infographic that I already put on the folder. So moving from the front to the back, that will be the how recent it is. The further to the front are the more recent pandemic, and the size of this dot represent the death toll. So the very recent one that you see at the front will be our COVID-19. So this was the latest infographic published by this website. It's called Virtual Capitalist. This was back in 2023. So the recorded death toll for COVID-19 then was about 6.9 million people. So if you go back in history, you'll see that HIV and AIDS that started in the 1980s has already killed estimated 25 to 35 million people. And that's how it works. If you can look at this later on in detail. This one I need a full screen to share, but never mind. Let me see. Okay, so they also prepare a death toll in terms of numbers. This is not looking at time frame, but this is looking at numbers of death so far. So the largest one is Black Death that we talk about, the bubonic plague that happened in the 14th century. It was predicted and calculated that to have killed about 200 million people in the past. And we also have plague of Justinian, and they killed about 30 to 50 million people. And we have the third plague, which is also known as Spanish plague, and this is 1855. So another image here. So what we're seeing here on the top left corner is one of those medical doctors. When they are the medical doctor who are treating patients who are suspected to be infected by the bacteria that causes bubonic plague. They wear a big light structures, and they actually stuff dry flowers and at the beak, because it is thought that the infections was spread through bad and dirty air. So that is what happened in the past. So they were partially right, not that 100% correct, but this, you can see that from what happened in the history, you can see the evolution of the health system and how scientists actually work their way to identify what causes infection. And also during the Black Death, it was first quarantine, the term quarantine, the practice of quarantine was first coined. So back in the 14th century, as I mentioned just now, a lot of people that came back from sea travel were found to be sick, so they weren't allowed to leave the ship until they are well. So that's also the concept of quarantine that we see today. Okay. Let me go back to my slide. Oh, I can do full screen. If I do full screen, you can see. Next slide. So in terms of the impact, as I mentioned, not just the medical and health impact, we also look at, so mainly on health and societal impacts, death tolls, increased understanding of disease, increased hygiene, washing of hands, and the use of the old concept of masks or barriers to prevent breathing in bad air, new health practices such as quarantine, economic disruption from the people who are infected weren't able to work during the period they were sick, or eventually death. And also social unrest, the example that I give you just now, the Jewish people, and also the religious practices and religion impact. So in looking at what the present day, one of the latest information published by WHO on the top causes of death, leading death today. So this is based on the numbers from 2019 to 2024. So over here, you will see different patterns here. If they're in blue, those indicate diseases that are communicable, mainly infectious pathogens. So on the list, top 10, we have COVID-19, we have number 5, lower respiratory infections. We also have tuberculosis. So from here, you can see that the top 10, at least 3 of these were caused by infectious diseases. So here in Malaysia, what is the scenario here? So in the past few years, whenever I look up on dengue cases, 2021, 22, 23, 24, 24, every year around the same time this year, April, it was always there was increase in the dengue cases. But this year, it was a little bit different. Dengue cases, there was a drop in Malaysia. So this is on the 28th of January. I couldn't find any newer news on the dengue cases. So on the 28th of January, it was said that even though the dengue cases dropped, but death rate is caused by dengue actually increased. So good news, and also a little bit of information for all of you. Dengue vaccine is now available. I think in Malaysia it started 2 years back. So if you would like to get vaccines for this dengue vaccine, there are 2 doses. Sunmed is charging $240 per dose. You would think that maybe Sunmed is more expensive, which may not be true, because last week when I went to a clinic outside, they are charging $250. So if you're interested, you can get this done, okay? And so what are the key things that is happening that is affecting us a lot in terms of medical microbiology? For me, I would think that it is the antimicrobial resistant AMR. So a lot of antimicrobials, antibiotics, antiviral, antifungal, antiparasitic drugs are now, what we're seeing is that they're losing effects against microbes. So in the past, we think that if you have infections, bacterial infection, by taking antibiotics, you'll be healed. But that is not always the case anymore because of AMR. And the AMR is also known as a slow motion tsunami. It is happened in the background. So AMR combines all bacterial or fungal infections and death numbers caused by the losing effects of antimicrobials. It is happening in the background. It is not like COVID-19 that it hit. You see a lot of people suddenly got infected, a lot of people hospitalized. AMR is slow motion. It's happening in the background at a rapid rate. So AMR can be caused by different bacteria, different fungi, different viruses, as long as they are not responding to the antimicrobial. So it is not as obvious as some of the pandemic that we see like COVID-19. And WHO has already said that the world is heading to a post-antibiotic era in which common infections, once treatable for decades, are now killing us. So when antibiotic was first launched many years ago in the 1960. These are some of the things that the poster that you will see. Thanks to penicillin, you will now come home. Penicillin cure gonorrhea in 4 hours. These are no longer that true because a lot of the microbes are now resistant against antimicrobial. So this is a rather old report that was published in 2014. It was predicted that death that it attributable to AMR each year by 2050, if nothing is done to control AMR, this is a number that we'll see. So for example, in Asia, we will be seeing about 4.7 million deaths in 2050. So that is the number that we're looking at. Of course, there are also reports and articles that are published to question the numbers that are presented here. For example, 4.7. So this is an estimate, just an estimate. But the key message here is, if let's say AMR is not controlled or monitored, eventually we will be running into a scenario where I have a sore throat and eventually the sore throat develop into secondary infections that can cost my life. Okay. So there are multiple things that has been done, global action plan, and multiple elements has been put in place. For example, it is now an urgent agenda that we must improve awareness and understanding of AMR. So I think all of you would have already learned about AMR, right? And also to conduct surveillance and research on antimicrobial resistance and to slow down the AMR. And another objective of the Global Action Plan is to reduce infection. No infections, less infection, less chances of resistant organism develop in you. So there's no infection, no need for antimicrobial, no death, no mortality affected. Okay, and also the other objective is to develop new drugs, diagnostic method, and also vaccine. And the last objective is to optimize the antimicrobial use in the hospital and in the healthcare system. So there are multiple things. I won't go into this in details later on. I don't know whether you're too overloaded by now. There's also Global Antimicrobial Resistance and Use Surveillance System GLASS report. So this is a surveillance approach effort initiative that's targeting the second objective of the Global Action Plan that I mentioned just now, surveillance and research. In terms of antimicrobial resistance in Malaysia, just a little bit of numbers. So Malaysia is also done, it's not done, I shouldn't say it's done, it's doing surveillance on AMR. So this is just a little bit of data on the common community. So in the community, meaning that these are people who are not sick. This is if let's say a person now you have infection, you go into the clinic or hospital, you get tested, and for example, In 2021, we're looking at the blue color here, 18% of the bacteria isolate were found to be resistant against Klebsiella, for example, okay? And also, if you look at another species, a very common bacteria species found in patients who are hospitalized is Acinetobacter baumannii. In 2021, 54 to 68% of these bacteria that found on patients in hospital are resistant against one of the group of antibiotic called carbapenem. And 42% of this, they are resistant against cephalosporin and sulbactam, which is very worrying. So if you, let's say you have Acinetobacter baumannii infection, half the chance is all this carbapenem, cephalosporin, sulbactam antibiotics will not work. If you don't isolate the Acinetobacter from your body, and if you do not do the antibiotic testing, and doctor give you this carbapenem, for example, 50% of the chances it will not work. So what happens then if it doesn't work? The doctor will have to put you on another antibiotics. And what happens to you? You don't recover. So that is the impact of AMR. Okay, antibiotics don't work. You get infection, you don't get treated properly. And more data on this, I will not go into too much of details of the AMR anymore. And if you think about this, what other infectious diseases are causing problem disease? So we talk about AMR, we talk about, what else do we talk about? AMR is one big one. And we also talk about the common ones, like what are the emerging new infections or re-emerging microbes that require attention as a medical microbiology student. So if you follow news, there are many cases that you will see, right? Report what happens here, measles coming back in the US. So what happened with the measles cases? Why is measles making a comeback in the US? Anti-vaccine, yes, correct. So things like that, I'm not sure, but in my view, they are totally preventable. So measles is one, but any other example you can think of. So I hope most of you would have gotten your vaccine, necessary vaccine, to protect yourself against infectious diseases. I hope. Okay, then let me see. Zika. Is there a vaccine? Oh, my connection is poor right now. Can you all still see me okay? Maybe seeing me as low resolution. Okay. Zika is the other one, is it? Slides, okay. Let me move on to my slide. It's a bit late now. All right. So I actually posted one review article, a super huge article. I don't expect you to read through all of them. I think there are 68 pages there. So, but these, why do I put this here? And the articles there. So remember we talk about assignment. So the emerging infectious disease may be one that you think about for your assignment. So emerging infectious disease is something that is new, that hasn't come out before. Like the new influenza, new different subtype that's making a lot of people sick. MERS-CoV is also considered as novel. They are not very, very new, but if you're looking at the time frame of like 10 years or a time frame of like 50 years, these emerging ones are quite new. 10 years, MERS-CoV is quite new. SARS-CoV-1, SARS-CoV-2, they are quite new. We're not talking about bubonic plague. We're not talking about the HIV that is quite old. HIV is 1980s, older than myself. I'm around that age, okay. And also we have different types: Ebola, Nipah, avian, Lassa, monkeypox. All these are new. Okay, and I also put in some tables for examples of the recent human emerging and re-emerging infectious diseases that you can use as a reference. If you don't read the full articles or you don't download the articles at all, you can have a check here. But do you think that we are all ready for all this pandemic? When 19 happened, I would think that we weren't prepared for the scale of pandemic that came, right? So how ready are we? So this is something that, for me, future direction, that is something that needs to be done. So moving on to future, so we talk about what in the past, what happened. We talk about now, what we are seeing now, emerging, re-emerging infectious diseases. We look at AMR. But future, what do we do? What is expected to be happening? So the technology, the new discovery are happening very quickly. So there are a lot of changes in the past 10, 20 years. So personalized medicine is one, for me, in my view, for medical microbiology. It's just like any other diseases. The infections that happens in individual should be treated, should be personalized. So one area, so what we're looking at is the patient's genetics, the immune response. Again, this is the effect of the patient's genetics, but also microbiome. This is what I see. The microbiome in different individuals varies. So I quote what is in the articles that I share here. These full articles are also posted on eLearn. The microbiome may modulate disease manifestation because of the microbe that you have in your body. You respond differently. Even among individuals with similar genetic disease susceptibility risk. So that's why in these articles that I posted, so this is one of the things that I think is for the future direction. The profiling of the microbiome in a person would also predict the outcome of specific treatments for specific infectious diseases. Okay, so that's my view of the future direction. And also, in terms of future, for scientists, lab scientists, likewise, maybe the rapid and point-of-care diagnostic is the way forward. So if you look at the conventional diagnostic method for infectious diseases, if it is bacterial infection, you want to identify what bacteria or what fungal species that causes infectious disease. It may take days and weeks to identify. Moving forward, and what is happening now is that the diagnostic should be very fast, should be on the spot point of care. It should be at the clinic, at the hospital when you go in and say, okay, I want to get tested. I want the result now. I don't want to wait for bacteria to grow. I don't want to test or wait for bacteria to respond to antibiotics for susceptibility testing. So the future diagnostic should be fast, cheaper, and portable. And it should be based on CRISPR, nanotech, and biosensors. So that's the direction at the moment for all R&D. And also, for me, in the future, microbiome research is very important. Like I said just now, one of the applications of microbiome profiling is for personalized medicines in the treatment of infectious diseases. But microbiome research itself is the understanding of what microbes that live on Earth. So we have already seen a lot of evidence that the microbiome that we carry, the bacteria, the fungi, even archaea or very little viruses that we carry have an impact on your mental health, your metabolism, your immune response to different foreign cell or pathogens. So the future treatment should also include microbiome transplant or engineered probiotic to prevent or cure diseases. So engineered probiotics is supposed to be meant to modify your microbiome to a healthier microbiome. So there are a lot of ongoing research in this area. I think it is very exciting. So at the moment, microbiology is no longer the conventional way. It should be more than just identification of bacteria, the study of one single bacteria in the human body and how does that causes infectious diseases. And the next thing that I think very important for the future direction of of medical microbiology is the pandemic preparedness. How ready are we against pandemic? So the future microbiology work should focus on early detection of new viruses. If you see the list that I shared earlier in terms of the emerging and re-emerging infectious diseases, many of them are viruses. Okay, early detection in viruses. And also we must work on the vaccine platform that can be quickly updated. So COVID-19 has changed how the launch of new vaccine in the history. So in the past, to get from the new vaccine development take 10 years. But now we know that it can be done very quickly and efficiently. So the global surveillance system for breakout tracking is also very important. Only if we can detect early cases very quick, we can be prepared for the pandemic that comes. Okay, and that's why recognizing the importance of pandemic preparedness, WHO also have this initiative called WHO Preparing for Pandemic. So the website is there. You can have a look later on. So in one word, what do you think? What do you feel is the biggest challenge for the future of medical microbiology? For me, I think is preparedness. So all of us have to work as a team. I wanted to do word cloud, but I think I'll skip that for now because we only have about 15 minutes. I don't know whether you have next class to rush to. So before we go, I want you to watch those videos that I posted on eLearn and read the articles. So there are only 2 articles I posted there. One is the utilization of microbiome for personalized medicines. I think it's very interesting read. The other one is the emerging and re-emerging infectious diseases. That is the 68 pages articles. You don't have to read, but you can just have a quick look. And in terms of assignment, as I mentioned just now, if you can, form a group quickly and select a topic. I have created a Zoom discussion page for you to just input your group members' names and also your selected topic so that I will have a idea what you want to work on. Is that okay? Let me stop sharing this. Yeah, is that all right? So before we go, just quickly, what do you feel that is the biggest challenge? I may have influenced you a little bit, but if you can give me a few keywords, I'll be very happy to see what you feel about that. What is the biggest challenge for medical microbiology in terms of medical microbiology? People scared of the technology. Are you referring to AI? Or antibiotics combination don't work? Imagine you have a sore throat, for example, and then the anti-vaxxer stuff. The vaccines are there for people to use. I agree, Jason. That's just not acceptable for me, in my view, personal view.